Bismillah ar-Rahman ar-Rahim. Assalamu alaikum wa rahmatullahi wa barakatuh. That's how we greet people in my part of the world, and that means peace upon you all. And I want to start by, does this work? Yeah, my name is Rana. And Rana, I'm sure many of you come from Latin America. That means the frog. But my mom didn't call me Rana because she was immured by frogs, because in Arabic, Rana means to contemplate. So I am the contemplating frog, and I hope you won't forget me because of that. But I also want to start by thanking Hilton for inviting me, as well as the rest of the team, Amanda and EA and everybody, because they were very persistent and patient in helping us make this happen. So it's not going to be perfect. We're learning on the way, but I'm very happy that we were able to be here, at least two of us, and the rest will be Zooming in. So thank you very, very much for the team and the Human Biology Association for inviting us. Now, I'm going to introduce myself slightly different way. I like to take control. And I like to say that I play many roles in my life. And this is a representation of my part of the world. And when I talk about roles, people talk about hats. I don't wear a hat, I wear a scarf. So I talk about my multiple scarves. And I start with my first scarf, is that I'm a parent, a mother. And to me, that's the most important role any human being can play in life, because you're bringing up the next generation. What could be more important than that? And yet we don't mention it and we take it for granted. And that's why I like to start with it. The second scarf I wear is I'm an educator. I was actually a school teacher for 10 years before I went and did my PhD. And of course, now at the university, I'm an educator. And I think the second most important role any human being can play is being an educator, because if the kids are not at home, they're at school and somebody has to take care of them. And the importance of the role of the teacher in forming and molding and inspiring future generations can never be underestimated. My third scarf is I'm a scientist. I get a kick and a thrill of trying to understand how cells talk to each other, proteins are expressed, and so on. So as Hilton mentioned, I work with Circassians and Chechens, but recently I've been working on the genetics and biomarkers of refugees in Jordan who come from multiple places. But I also, as he mentioned, work on the Chechens and Circassians who have settled in Jordan as well. My fourth scarf, before I go to the fourth, of course, working with stem cells in the Circassians and the Chechens, when we discovered a novel gene risk factor for diabetes, made us work with stem cells to express those genes, because nobody knows their function. And that led me to actually dip my hand into working in policy, because we didn't have a stem cell research guideline or for therapy guideline. So, when it's not there, you have to step in and make it happen. So I created a team of multiple stakeholders, and we created the first stem cell law in Jordan that became kind of a guide for other countries in the region. So you go places you didn't imagine. My fourth scarf is I'm a social innovator, and that's why I started the program We Love Reading, which I'll talk more about in my talk as part of the 6 talks in our place-based session. Now, I mentioned 5 scarves. I usually don't tell you what the 5th scarf is, and I invite you to read my book, which I'm going to donate for the raffle for next year. So that's for next year. It tells my story, and you'll read it and guess what the 5th scarf is, or you will maybe by the end of this talk. Now, the other thing that I talk about, where I come from. So I'm from Palestine. My father is from Jerusalem. We've been in Jerusalem since the 11th century. This was the last picture that was taken in 1942 of the whole family. We have a whole neighborhood. And in this neighborhood, we lived with Christians, Jews, and Muslims living side by side, working together, building a thriving civilization. However, in 1948, my dad was 5 when the Zionist forces evicted him violently with the rest of the family to Lebanon, and then later, like many in diaspora all over the world. And unfortunately, this diaspora is happening again with the genocide unfolding in Gaza. So trauma is in my DNA, if you will. Now, today, this is the 50th anniversary. I'm so lucky that our place-based session was at this anniversary at this time. And I loved the over the horizon sessions and the discussion last night about how can we look at the future and what are we going to do and how can we do better science for humanity? Because in the end, that's what we're doing, science for humanity. So I have a few suggestions that come from my part of the world. So first of all, what can we do about the past? Right? Because science, we need to decolonize and uncolonize science, or whatever the right word is about doing that. And the first thing is to acknowledge those from the past. So Al-Bayruni, who was a scholar, is recognized as probably the forerunner of anthropology. And I don't know if the human biology book talks about him or not. I haven't read the book, but I think it should be, because this is about reclaiming the scholars of the past before Western civilization and the Enlightenment, because people were enlightened all over the world. So we can fix that by talking about these scientists and scholars and sharing that knowledge and that wisdom, because we need it more than ever, the diversity of knowledge and experiences. But what about the present? So I wanted the present in my region is that there's a genocide happening. The International Court of Justice already issued a statement that this is a genocide. Many Jewish scholars, experts on genocide and survivors or descendants of survivors of Holocaust have said that publicly, yet it continues, even today. And if we want to look at it as scientists, the UN has called it a scholasticide. The data, the students, the teachers killed, the universities destroyed, and the professors that have been killed are more than we can know. As a Palestinian original female scientist, I was following my female scientist colleagues throughout the years under oppression since 1948. And we published actually in Nature describing their life of how do they do science under occupation, under apartheid. And actually, one of our speakers today is going to be calling in from Palestine to talk about her journey and her research in Birzeit and Nablus. But many of those colleagues that I followed actually were killed with their families. And today the count is 140 academics killed with their families in Palestine, in Gaza in particular. And I ask for a 30 seconds moment of silence to honor our academics and our colleagues. Thank you. So when I came to the conference this year, I remember I came to the HBA in 2022, and it was when Russia had invaded Ukraine. And everywhere in the conference room in Denver, there were blue and yellow flags, blue and yellow bracelets, stickers, pins, all over. But when I came this time, there was nothing about Palestine, nothing. I was the only one wearing this as a representation. And I just pose a question mark. Why? Aren't we all equal? Aren't all academics deserving of being recognized? And that's something to sit with us, to think and reflect. What is our duty as scientists? And it's not that just, some may say it's about Palestine, oh, that's too far away, it's across the ocean. But all oppression is connected. We know that. We know that as scientists. Black Lives Matter, what's happening to the Black African community in the US is connected to what's happening in Palestine, because it's about structural barriers and racism. Indigenous communities fighting for their own rights here in the United States is connected to the struggle in Palestine. And LGBTQ people who are fighting for their rights is connected to what's happening in Palestine. And we can learn a lot from colonialism, and I'm sure you're familiar with Frantz Fanon and his books and his philosophy about the French colonization of Algeria. So we're all connected. And it's not about religion. Religion is used because there are so many Jews, Voice for Peace, who are working together with Palestinians and Christians to uphold justice and to call for justice and accountability. So we're all connected. And I don't want to wait, because I've been to many conferences where they celebrate the indigenous people. Today we talk about the land we're standing on that belonged to tribe X and Y. I don't want to wait 100 years for somebody to come stand in Palestine and say we acknowledge the land that belonged to the Dajani family. I want to do something now. What can we do now that we're aware? We know what happened, and therefore we have a responsibility as scientists. Even though we may say, I'm a scientist, I have nothing to do. No, we all have a duty. And as Einstein said, the world will not be destroyed by those who do evil, but by those who watch without doing anything. So we have a responsibility, and silence is complicity. And what can we do? So we can do boycott, divestment, sanction. That's not violent. That's a way to show solidarity, to break the power balance, to hold people accountable where it hurts, which is money and power, because it's all about money and power. And it's all connected. Even climate change is connected, because climate change is about corporations who take advantage and who really don't care about humanity. And the only way to stop those who are impacting climate change is also through boycotts. And as a famous book that worked on climate change, it takes a lawyer, an activist, and a storyteller to change the world. So these are things we can learn as scientists. How can we use our science and our voices to make a difference. So storytelling is very important. And that's why I imagine as human beings we're evolving, I imagine this. There were two groups of people. There were a group of people sitting around the fire telling stories, and there was another one that frowned upon them and accused them of wasting their time. But you can guess which one survived. It was the storytellers. So we need to be good storytellers. And this is I'm a co-founder of this initiative called Act for Palestine, and it's an academic network for Palestine about raising awareness to decolonization, what's happening, what can we do as academics to support each other across the world. So you can scan the QR code and hopefully join us and be part of that movement. And it's all connected. So when you help what's happening in Gaza, you are helping what's happening in the United States and fighting for everybody's rights. So that's for the present, what we can do. Now, what about the future, right? What can we do? And I mentioned yesterday systems change. How can we imagine a new system to replace the system that is not working? And I'm going to give you an example of how that can work if we all work together. And the example comes from my other side of my family. So my mother's Syrian. She's from Aleppo and Hama. And that's the history of the whole region. We're all connected, right? There were no borders. It was all the Levant. So we're all married from each other and amongst each other. So in 1982, in February, so this is the anniversary just passed this last month, there was a massacre by the Assad regime, and around 40,000 people were massacred. Many of them were from my mother's family. And in December 8, 2024, the Assad regime finally fell. And people were celebrating in the streets. But because of the intricate history of my mother and knowing what happened and being a scientist, curious about trauma, because I am, again, the descendant of both sides of my family. And when people talk about trauma, they talk about one event of trauma, when I know it's a continuous event. And how do you study trauma when it's not just one event? And because of knowing these families, I asked the question, can we trace an epigenetic signature of trauma across 3 generations in humans? We know we've done it in animal models, but nobody's been able to do it in humans because ethically you can't. But you can retrospectively if you know the society and population, if they trust you. And actually, this is a picture of 3 generations who were part of the sample. So we took direct exposure, the grandmother of the 1982 massacre, when she was pregnant with the prenatal, and then we had the germline exposure, who later was born either before or after the 2011 war. Now, this was not an individual effort. I started the question and the idea, and then partnered with Catherine Panterbick at Yale, who helped provide funding and assessment of what is trauma, because she's an anthropologist and I'm not. And then with Dima Hamadmad, she organized collecting all the samples, which took over 7 years, going around finding these families all over the world. And then when we got the samples and the DNA and shipped them to Florida, to Connie Mulligan's lab, who is an expert on epigenetics, where she analyzed all the DNA and did an EWAS, and Alexandra Binder from University of Hawaii helped in this. And then together, we were able to discover, with NSF funding, 14 sites of epigenetic signature that were inherited by the grandchildren from the grandparents. And these grandchildren were not exposed to the war. Now, and we published this last week in Scientific Reports. And it's groundbreaking in the sense that it's the first time in humans. Now, what does this mean? So what it means is that these 14 sites are not in any known biological pathway. So they're not the typical cortisol pathway of stress and so on. So that gave us an opportunity to take a step for systems change. Can we imagine what that could mean for future generations? That you have higher regulatory sites affected by the trauma that allows the future generations to have adaptability and flexibility in dealing with an unknown, unpredictable future. And that's why we coined the term, not victimhood by vulnerabilities, but agency through adaptation. And what does that mean in terms of doing better science when you have this framework of looking at the experiences and phenomena? How does this impact intervention design, intervention measurement, policy, et cetera? And what are the implications for the families of this discovery? Again, bringing it full circle. We went back and I actually visited one of the families last week who had given us a sample 10 years ago. The granddaughter was 2 years old. Now she's 12 in school and she wants to be a physician. And we sat with her and explained all the research and what happened so that they understand that what they contributed made sense. And they were so happy because when they gave us the samples, they were hush hush because they were scared of telling their stories of torture and imprisonment. But today, because the regime had fell, they were able to publicly talk about it and then use our data to hold those perpetrators accountable, and having now the agency with real biological evidence, but also to call for stopping the violence altogether. to start from the beginning, to stop the source of all of it, and also giving them hope for resilience for future generations, rather than being deterministic that, oh my God, you're victims. And that's doing better science for humanity. And this is an example of teamwork among a group of female scientists across the world, each one pulling in to have a collective discovery in the end. So the place-based session. So it's about the Middle East and North Africa, and I gave you an overview of what this session is about by sharing what I shared. As you can see, it's multiple countries divided by colonizers, but it was all one region in the past. We share language, we share history, we share culture, and we share a lot of civilization. I checked quickly. There was a study that came out in 2023 about the research coming out of the region, just to give you an input. And this shows per country the scholarly outputs relative to GDP. And as you can see, per year, by country from 2003 to 2023 or 21. And you can see that I think Iran is one of the highest, and then Egypt. But then if we look at the scholarly outputs by year, by region, collectively comparing the European Union, the Middle East, and North America, obviously, the Middle East is way at the bottom, the red line. But if you adjust for per capita, then the Middle East is actually doing very well, as you can see in the bottom graph. So there is hope. And we can do more. And that's why data analysis is very important, and not to do stereotyping, because one could say, oh my God, they're doing terrible. But when you adjust, you think, no, they're doing very well. So be careful of the stereotypes that come out of the media, and always ask for data and evidence. And also this also shows the percentages of international and national collaborations by region and by country, comparing the Middle East with Europe and North America in the first graph, and then below in per country. So these are some data to show that we can work together locally and internationally. And then lastly, there's always the stereotype that we don't have enough females in the MENA region who study science, and that is wrong. That's a false assumption propagated by media. We all know the fake news, we're all familiar. UNESCO actually produced a report that 57% of science, technology, engineering, and mathematics graduates in the Arab countries are women, which is better than the US or Europe. So maybe there's something anthropology can learn from us. We're doing something right. And let's do that mutual didactic learning from each other. And one of our speakers from Palestine is actually featured here in that picture from UNESCO. She's the one in the middle with the red scarf. So we'll be hearing from her later. With that, enjoy the show. Our second speaker is Faten Atrouz, and she will be speaking about metabolomic insights into the biochemical underpinnings of stress. The floor is yours.
Good morning, everyone. Thank you for joining us today in person or virtual via Zoom. And first, I want to acknowledge this opportunity, and I want to acknowledge all the presentations and the posters presented in this talk, which reflects like amazing research conducted by this community. I'm happy to be part of this meeting this year. I'm excited to share with you my research entitled Metabolomic Insight into the Biochemical Underpinning of Stress. This is a collaborative studies conducted between our lab at the University of Houston College of Pharmacy here in the United States, and with collaborator from Jordan University of Science and Technology at Jordan, and with our also collaborator from the University of Sharjah at United Arab Emirates. I will start by asking you a question. Who claimed that he never experienced any stressful life event? None, right? This is part of a human life, right? Yeah, well, like stress is essential for our development and growth, right? But the case is different for chronic stress, right? Chronic stress is the effect or detrimental effects of a chronic stress is well documented, and its impact on brain development and its association with psychological disorders is also well documented, right? So there are several metabolomic pathways that suggested to underline the connection between stress and psychological disorders. And in fact, our preclinical studies that conducted on animal models of stress connected oxidative stress and inflammatory markers with chronic stress and disorders. Also, there are several studies conducted by other researchers that also propose the same or similar metabolomic processes or cascades, which is related to oxidative stress and inflammatory marker. So the aim of this study is to examine the salivary metabolomic profile of a highly stressed population or a group of population who exposed to chronic stress and compare it to another population who are not exposed to chronic stress as the and that group is the Syrian refugees. I want to talk to you a little bit about the Syrian refugees. Syria in the Middle East here has a civil war in the 2011, which forced around 14 million of Syrians to flee their home searching for safety. And around 6 million flee to the neighboring countries, including Turkey, Lebanon, Jordan, Iraq. And around 600,000 Syrian refugees resettled in Jordan. These are the registered refugees. There are more than 1 million non-registered refugees that just passed the border. It's like walkable border between Syria and Jordan. And it is well documented that refugees are at high risk of for developing psychological disorder, which is due to combination of a pre-migration trauma exposure of war and terror, and the stressors correlated with forced displacement, and either also the challenges that they face after resettlement in the host country. So we targeted Syrian refugee, and in this study, we specifically targeted refugee women, Syrian refugee women. Why we targeted Syrian refugee women? In fact, this study is part of several studies that conducted with Syrian refugee who resettled in Houston, USA, and Syrian refugee who resettled in Jordan. And in these studies, we explored we examined their mental stress, their perceived stress, their mental distress, and we also measures like the conditions or the context of resettlement and the conditions of resettlements and how it's connected with their mental distress or perceived distress. And in all these studies, we found that female reported higher stress and distress as compared to males. So we decided to focus on this group of Syrian refugee, the Syrian refugee women. So this is a cross-sectional studies that aim to identify changes in salivary metabolomic profiles of Syrian refugee women and compare it to non-refugee Jordanian women. This study was conducted in the northern part of Jordan, this part of Jordan, which is like right across the borders with Syria. This study was conducted in between July and October 2022, and it included adult Syrian refugee, Jordanian, and women. The sample size was 100, around 50 participants per group. The data collection was interview-based data collection, and the survey included socio-demographic data and psychometric measures. Psychometric measures that we used in this study included the validated Arabic versions of the self-reporting questionnaire and the perceived stress scale. The self-reporting questionnaire assessed the perceived stress reported by participants, and it composed of 20 questions, 14 questions, and 20 questions, and perceived stress composed of 14 questions, and it's like evaluate how much they handle the stress and how much they expose to daily life stresses. Saliva samples collected from the participants using the passive drool methods, and this saliva sample used for the metabolomic analysis. Metabolomic analysis was conducted at the University of Sharjah at UAE in the lab of Dr. Azabi and Dr. Samirin. A global quantitative metabolomic analysis was performed utilizing high resolution spectrometer TIMS-TOF, and the Metascope for software was used for data analysis. A total of 146 metabolites were identified using this method, and the human metabolome database was used to identify metabolomic pathways that are differentially regulated between Jordanian and Syrian refugee women. So this table shows the socio-demographic characteristics of participants, and as you see, education level. As you see, education level is significantly different between Syrian refugee and Jordanian women, with Jordanian women reported higher education level as compared to Syrian refugee. Employment rate was higher among Syrian refugees, but income was very limited in Syrian refugees' families, with average monthly income less than the poverty line, which is around 351 to $775 per month. Also, they reported that they do not have health insurance, but the other body markers like body mass index and the chronic disease was comparable between the two groups. For the psychometric measures, this graph shows the results of analysis of psychometric measures in perceived stress scale. Actually, both groups reported high perceived stress, given the situation in the Middle East and the political turmoil and the genocide going on in the Middle East. As Dr. Dajani said, this has impacted the stress of all individuals, I think, in the country, in the Middle East. So both actually reported higher than the average stress reported by people, which is around the average reported score for stress, perceived stress is 20 to 23, and here both groups reported 27 and 30, the average score, which is significantly higher among Syrian refugees women as compared to Jordanian women. In the self-reporting questionnaire, the differences between the scores of Jordanian women and Syrian refugee was not that high, but there is a difference which is higher among Syrian refugee. And when we further analyze the subscales of self-reporting questionnaires to include two subscales, which are anxiety symptoms and depressive symptoms, we found that depressive symptoms are higher among Syrian refugees as compared to Jordanian women, while anxiety symptoms was compatible between the two groups. For metabolomic analysis data, the analysis showed that around 29 metabolites were upregulated in the saliva samples of Syrian refugees. Further enrichment analysis shows that these metabolites are belonging to the amino acid and the purine metabolism pathway and energy regulation pathways. And these are upregulated in Syrian refugees as compared to Jordanian women. So to conclude our study, we found that Syrian refugee reported higher perceived stress and higher depressive symptoms as compared to Jordanian women. And both Syrian and refugee women scored high in self-reporting questionnaire. And amino acid, metabolomic pathway, and neurotransmitter precursor, specifically the neurotransmitter precursor amino acids pathway are mostly altered in Syrian refugees as compared to the Jordanian women. So these amino acids, precursors of neurotransmitters, their expression is highly correlated with oxidative stress and inflammatory markers. So these expression or metabolic cascade of these amino acids, degradation or synthesis of the neurotransmitter is impacted highly by these oxidative stress and inflammatory cascades. So in our future studies, we will focus on these pathways and to study how specific biomarkers within these metabolic pathway is correlated with mental disorders, clinically diagnosed mental disorders in highly distressed individuals, not necessarily refugees, but highly distressed individuals who experienced lifetime trauma. And finally, I want to acknowledge our team, the PI of this study, Dr. Salem, and our collaborators from Jordan University of Science and Technology in Jordan, our collaborators in the United Arab Emirates, the funding mechanisms, and the women who participated in this study. And this is our picture in Jordan in summer in 2022. And thank you.
So we have a minute or two that they're testing the sound for the next speaker. And we're going to go back to Delaney, the first one. But meanwhile, we have time for questions. So if anybody has a question for Fatin, there's a mic there in the back. Please ask, or a question on the previous one with the introduction to the place-based session. Please, and we want your, not just your questions, your critiques, because that's how we all grow together when somebody critiques us.
Hello, I just want to ask Fatin, how pervasive is mental health disease in the refugee population?
Yeah, that's a great question. Actually, I directly contacted with Syrian refugees and I conducted all of the research or interviews conducted here in the United States and in Jordan. Actually, initially, these people like trying to hide it. But when you start talking to them, especially when we are from the same background, same culture, the session ends with tears from both of us. Like they are really, really like hard experience, like with their families, with the seeing like loved one missing someone's, how they survived the war and the displacement. It's like heartbreaking stories, yes. And they try, the problem with these people like try to hide it. They do not explicit about it. And that's what's more painful. Like I feel these women like heroes because they survive all this pain and start trying to hide it specifically like from their families, from their children, doing their best to have everyone is happy in the family and while they are suffering in silence. Like some of women told me like, oh, it's the first time someone like care about asking me these questions, like, and she started crying and just telling some stories. Yeah, so it's very hard. Thank you. Any other question? Yes, please.
Thank you so much for that wonderful presentation.
Thank you.
I remember from Rana's slide that you were working with Professor Catherine Panterbrick, who's a good friend and colleague of mine. And one of the things that my question to you is drawing from her research in Afghanistan about different types of stress that families face. So for example, from her work, and I think in Kabul, was how stress in the family manifested itself in different ways from the stress incurred by wartime violence. And I'm wondering if you saw anything in your research in terms of how different types of stress may affect your research partners in different ways. Thank you.
Thank you for the question. Actually, like we did not dissect like different types of stress, but we have notes about these stress. But in general, like degree of stress reported associated with high scores, like how much they reported stress associated with high scores in general. But we have these like, qualitative data that needs further analysis, which might be like a great for future analysis, it's a different type of analysis. Thank you for the question.
Hi, my name is Delaney Glass. I'm an assistant professor at the University of Toronto, and I'm excited to talk to you today about hormonal coupling. Before I get started, I just want to acknowledge the genocide in Palestine and the effect that's had on so many personally and professionally, and I'd like to dedicate this talk to my colleague and friend, Doa Rabah and Aisha Noor, who actually were killed during the genocide. To position my work, I focus on adolescent populations in Jordan and Toronto, pictured in the middle and the right. And I'm a biocultural anthropologist. I'm really interested in the effects of early adversity on adolescent growth, development, and health, and in particular on mixed methods research integrating qualitative, quantitative, and biomarker methods. I focus on adolescence because it's a sociocultural transition, roughly ages 10 to 24, but also includes really cascading events of puberty with changes in height, lean muscle mass, hormones, and brain development that confer really a sensitive period to study growth and developmental changes. As a reminder, on the left hand of the screen, there's the relationship across age to different pubertal events. There are many things going on in puberty. This is just a reminder that menarche is occurring relatively later. Changes in hormones and other secondary sexual characteristics occur kind of throughout and across. But this is just a reminder that there's a lot of variation in pubertal timing and tempo. There are somewhat global trends towards relatively earlier pubertal timing as pictured here. The lighter colors represent trends towards earlier puberty, and the darker colors represent relatively later puberty. In the context of Jordan, less is really known, and I would say this is similar across the Southwest Asian, North African region. There's not a lot of studies happening that are documenting these changes. In Jordan, it's unclear if there's been a secular downward trend in age at menarche or in puberty overall for boys and girls. Some studies from the 90s as well as more recently show that the median age at menarche tends to be around 13 or 14, and it's quite variable therein. My work on puberty really tends to focus on the mechanisms that might be implicated in the relationship between early adversity and earlier or later pubertal timing. So this is a figure from my 2022 publication in Evolutionary Human Sciences about the contributions of psychosocial and energetic factors on pubertal timing. And I want to draw your attention to the bottom, which is just looking at the mechanism of neuroendocrine regulation between the hypothalamic pituitary adrenal and gonadal axes. This is one of many potential mechanisms that is permissive in the timing of puberty. These two axes have endpoints of testosterone and cortisol, and both are involved in response to challenges and stressors, and may kind of co-upregulate in puberty specifically. Cortisol, as many of you are aware, is released in response to physiological challenges, as well as being involved in energy use, body fat metabolism, and blood sugar. And it's been used really dynamically in the past. However, less is known about how early life stressors and developmental changes like puberty relate to cortisol and changes in cortisol across the adolescent transition. On the other hand, testosterone is kind of well known to increase across puberty. It's produced in the testes and ovaries and is involved in increasing bone mass and density, development of the gonads and secondary sexual characteristics. And although it was studied as sex-specific in the past, we know there's appreciable levels in both males and females. And it's also responsive to social ecological challenges. So if we think about standard pubertal growth curves, these are pictured by Ellis in 2017. These are not real data, these are synthetic. I just want to draw your attention to the age range of 10 to 15. We can see that everything but the blue, which is growth rate rather than a hormone, is increasing and then kind of reaches a peak and then decreases as an adult phenotype is attained. So we can expect that a similar trend might hold true for both testosterone if we were to map testosterone on here, and maybe even cortisol if we were able to look at the developmental trajectory of cortisol. And we might expect that the relationship between these hormones may follow a similar trajectory at the population level across puberty. In order to investigate this relationship, I use a concept called hormonal coupling, and this is the association between the HPA and HPG levels or changes in hormone levels over time. And generally, prior research finds that there is positive associations between cortisol and testosterone across the day, which makes a lot of sense given that there are diurnal rhythms in cortisol and testosterone. And some research finds that advancement of pubertal status is also related to this sort of heightened association or relationship between cortisol and testosterone. There's an overarching developmental hypothesis around coupling, which is this idea that, again, if we think about these pubertal growth curves, perhaps this relationship between cortisol and testosterone being co-upregulated to meet the developmental and energetic and psychosocial stress demands of puberty, perhaps that relationship is detectable at the population level in a large mixed age group. So just some terminology: positive and negative coupling refers to the direction of an effect, weak and strong is the magnitude, and decoupling is coupling that gets weaker over time, age, or development. And we think that this could be one of many ways to proxy really pubertal timing at the population level. If we can kind of see these trends in hormones across age, we might be able to map that onto pubertal timing and stage. However, there's really a paucity of research on hormonal coupling outside of Western contexts. We know that hormone levels and physiological patterning may differ in majority world context, where a majority or 85% of the world lives outside of the West. And we know that the effects of socioeconomic marginalization specifically and the extreme stressors encountered during displacement and armed conflict may have distinct roles or distinct effects, or more detectable effects rather, on hormonal coupling and adolescent developmental trajectories. So we think it's worthy of investigation outside of minority world or Western contexts. The Syrian crisis displaced 1.3 or more million refugees into Jordan, over half of whom were under the age of 18. And in Jordan, Syrian refugees face a complex life living in and out of refugee camps. They have differential exposures to poverty, as well as prior exposures to conflict. And then Jordanian non-refugee counterparts living in northern Jordan also experience their fair share of marginalization and heightened psychosocial stress, although different, of course, not being as exposed to sort of conflict-related stressors. To understand how these different stressors may have impacted hormonal coupling among Jordanian non-refugee and Syrian refugee teens, I worked with the Nubadr, or Advancing Adolescence Study in Jordan. This study was conducted by Dr. Catherine Panterbrick and Dr. Rana Dejani, 2015 through 2017, and the study represents around 817 Jordanian non-refugee and Syrian refugee teenagers ages 10 to 19. They participated in a randomized control trial, and the study was really evaluating the effect of the intervention of a psychosocial reduction intervention on these teenagers. So they collected data at baseline pre-intervention, at 12 weeks right after the intervention, and at 1 year later. My analysis is concerned with baseline and 1 year later. Broadly, they collected many different subjective and other markers, but I'm focused on insecurity, distress, perceived stress, mental health, resilience, trauma, and household wealth. And I worked with the University of Washington Mass Spectrometry Center to assay free cortisol and free testosterone in dried blood spot samples from around 700 Jordanian and Syrian teenagers. This is a photo of the mass spec lab at University of Washington. For statistical methods, I'm happy to talk about this later, but we used Bayesian regression models using the R package in law with around 700 participants at baseline. And our main model of hormonal coupling was to look at the effects of age group, cortisol, and an interaction between age group and cortisol on testosterone, and this is referred to as an age-specific effect of cortisol. This was used to capture a potentially nonlinear relationship to be able to capture both coupling and decoupling. We stratified by domains of stress and trauma, and we used a random walk-to prior to share information across age groups and to be able to more robustly detect these effects within and across age groups. To briefly outline what coupling might look like, this is not real data, this is just me showing you kind of the conceptual model that I started with before even analyzing the data. We're interested in understanding what is the age-specific effect of cortisol and testosterone to try to capture this kind of developmental hypothesis that we could see across a mixed age group, right? So at age 8, starting on the left, we wouldn't really expect a relationship between cortisol and testosterone. These hormones are just coming online, and we would expect no robust relationship. And then as age increases, and we expect varied kind of pubertal stages to be occurring around this time, we would expect a positive progressive coupling relationship between cortisol and testosterone. And then as an adult phenotype is attained and as puberty is kind of tapering off, we would expect a weaker, flatter relationship, and this would be kind of evidence of decoupling. So we predicted overall that the age-specific effect of cortisol on testosterone would be positive at younger ages and become decoupled at older post-pubertal ages. We also thought that this would differ for females compared to males because females are generally having puberty earlier. So we did find some evidence of positive coupling for males and females. When we see males in pink and females in red, the relationship is stronger across age for males, and within ages, of course, there's a lot of overlapping of the credible intervals, but we do see kind of this trend in coupling across age. Next, we looked at differences by nationality, which was representing kind of refugee versus non-refugee experience. But it's important to kind of give you some context for differences in overall levels of these hormones. So for Syrian females and males, they had greater overall levels of testosterone and cortisol compared to Jordanians. Syrians also had a greater magnitude of change in cortisol compared to Jordanians. But there was a similar magnitude of change in testosterone for both Syrians and Jordanians overall. So kind of consistent with the hypothesis that greater psychosocial stressors and traumas may accelerate pubertal timing, we predicted that Syrians compared to Jordanians would have sort of more positive coupling at earlier ages and earlier decoupling. We found mixed evidence for this when we looked at Jordanians in orange and Syrians in pink. You can see that there's a stronger coupling relationship for Jordanians as compared to Syrians. We also thought that maybe greater lifetime trauma as well as insecurity and distress would show a similar relationship indicative of earlier pubertal timing. For females, we found that there was still a detectable hormonal coupling relationship for Syrian females with greater lifetime trauma, but not a lot of differences between age groups. We found a similar relationship for Syrian females with greater insecurity and distress, but there was sort of weaker coupling at older ages. So this is just a preliminary line of evidence for decoupling, but it's quite weak at that. For males, Jordanians and Syrians with lower lifetime trauma events had a more robust coupling relationship, and there was also greater changes across ages. There was no robust relationship for insecurity and distress, and we looked at mental health, resilience, and household wealth, and we didn't really find much of a relationship to coupling. Overall, we found some evidence of positive coupling, no evidence of decoupling, and we suggest that maybe this is not a universal trend, but rather context-specific. We also think that different patterns and exposures to stressors may point to differences in pubertal timing, but also earlier life kind of hormonal modulation or integration. And future research should account for body fat, metabolic rate, as well as individual variation in pubertal stage. In the future, my lab is working on projects around social safety and well-being, following up on work in Toronto, and then we're also following up on hormonal and mixed methods research on multi-generational changes in teenhood. And again, this was following up on community feedback from last year in Jordan. And I would love everyone to check out the work that my lab's doing at University of Toronto and reach out if you're interested in collaborating. And thank you to the funders as well as my co-authors.
Our next speaker is Youssef Edgour. He's a professor at NYU Abu Dhabi, and he's going to be speaking about the impact of lifestyle on immune function and stress biomarkers in the Moroccan population. He's joining us online, and there's a recording, I think. And of course, I'll remind everybody, keep notes, questions, and we'll have a discussion at the end of the session.
Good morning, everyone. Today, I will present some of our work on how lifestyle transitions and particularly urbanization impact physiology in the Moroccan population. My name is Youssef Edgour, and I'm based at New York University in Abu Dhabi. So throughout history, human societies have undergone significant lifestyle changes. One of the most important transitions was the Neolithic Revolution, which marked the shift from hunter-gathering to settled agriculture about 9,000 years ago. More recently, rapid urbanization has contributed to the rise of complex diseases such as asthma, cancer, depression, anxiety, cardiovascular, and cardiometabolic disease. And these transitions in lifestyle align with changes in diet, exposure to pollutants, and psychosocial stress, among many other environmental exposures. My research examines the molecular and genetic responses to these lifestyle shifts. And while our genetic makeup is important, environmental influences such as exposure to different microorganisms, including pathogens and the microbiome, different geographies, diet, pollutants, stress, as well as many other factors, can be way more important in the context of triggering complex diseases. So to study these effects, we are conducting a series of studies in Morocco, and the Moroccan population is highly genetically diverse, with the Amazigh people referred to sometimes as the Berber people being the native population of North Africa, with contributions from other populations like the Arabs or Sub-Saharan Africans, as well as other populations. All these populations contributed to the modern genetic makeup of the population. Similarly, Morocco is a country rich in terms of geography and lifestyles, and here I'm showing the three main lifestyles that have been subject to our research in the last few years, namely the typical urban lifestyle, shown here in the coastal city of Agadir, traditional rural lifestyle in the villages of the Atlas Mountains, and as well as the desert nomadic lifestyle with only a few thousand individuals living these lifestyles lived in Morocco. So in the last few years, we recruited approximately 400 study participants. We collected blood samples, as well as questionnaire data on diet and other variables. We also done or collected blood chemistry data from the study participants. So in my talk, I will focus on the comparison between the urban and the rural lifestyles. So we generated whole genome gene expression profiles with approximately 22,000 transcripts being expressed in the blood of these individuals. The origin of these RNA comes from mostly circulating immune cells in blood, such as monocytes and lymphocytes. And we then conduct differential gene expression analyses to identify genes that are differentially expressed between these lifestyles and locations. So first, we compared the profiles between the two urban sites shown here, Cheira and Anza, and we detected no statistically significant differences in their gene expression profiles, so we decided to combine them as one group. On the other hand, we kept the two rural villages separate. So considering that we had individuals who self-identify as Arabs or Amazigh, we tested what would be the impact of self-reported ethnicity. So we compared the two ethnic groups within the same environment and detected no statistically significant differences as shown in these volcano plots. So this clearly demonstrates that the impact of genetics, or at least self-reported ethnicity, is minor. And so next we wanted to see now what are the differences between the different lifestyles or geographic locations. So we do perform the same analysis. In this analysis, we combine the two urban sites, as I mentioned earlier, and then we compare the urban site to each of the two rural sites, and we also compare the two rural villages to each other. And overall, as you can see in these volcano plots, we detect a strong impact of lifestyle on gene expression, with approximately a third of the genes expressed in blood being differentially expressed, around 7,000 genes out of 22,000 genes. However, we noticed that the number of genes differentially expressed between the city of Agadir and the village Ighrim, as shown here in the middle panel, was way much lower than the other comparisons. Well, it turned out that when we stratify our analysis by sex, we recovered the strong effect of lifestyle or location in females, but interestingly not in males, meaning that males in the Ighrim village have gene expression profiles similar to the males who live in the city. It turned out that most of the males who live in this village actually commute every day to the neighboring city, and this pattern in their lifestyle was sufficient to make their gene expression profiles similar to the males who actually live permanently in the city, demonstrating again a very strong sex by environment effect as opposed to a pure sex effect. So in this heat map, I'm showing the top 1,399 most significantly differentially expressed genes. These are all the genes that are significant at Bonferroni levels of significance. And this is between the urban lifestyle or the city of Agadir and the Botros rural village. And the red indicates higher expression, and blue shows the lower levels of expression. Each row is one gene, is one of the genes, and each column is one individual. So you can clearly see that we tend to see an increased levels of levels of expression in the city, in the urban lifestyle. So to make sense of these differences, we performed pathway enrichment analysis, and the results show a very clear enrichment of disease biomarkers in the urban lifestyle. We're talking about biomarkers of cardiovascular disease, inflammation, as well as related conditions. Next, we want to investigate gene expression biomarkers of stress specifically and compare the two lifestyles. Here I'm showing a map of the human genome and all the discoveries reported from the genome-wide association studies made by the year 2009. By 2012, notice the increase in the number of discoveries that have been made. These discoveries reported specific genetic variants as well as genes associated with multiple diseases and conditions. And since then, thousands of GWAS studies have been conducted, reporting hundreds to thousands of variants with varying effect sizes, of course, in different populations and for each disease and phenotypes investigated. So when we mined the GWAS datasets, we found over 1,000 genes that were robustly associated with behavior in general, but specifically with stress and related mental health disorders. Now, these conditions or these disorders encompass a wide range of conditions, as shown here in this figure. Our focus was mostly on common disorders such as depression, anxiety. And here is a Manhattan plot from one of the major GWAS studies that reported genes and variants associated with 5 major psychiatric disorders. And again, this is including depression. So given that our study's focus is gene expression, the basic idea is that the majority of these reported genes or genome-wide association signals might actually be acting through regulation of gene expression. Other mechanisms, of course, like epigenetics, will of course have an impact. So in our data, we extracted 371 of these genes that are expressed in blood. So out of 1,000 genes that have been reported in GWAS studies, 371 are expressed in our study participants. And then we conducted unsupervised analysis using principal component analysis. The idea of this analysis is to look at the gene expression profile specifically associated with stress and see how individuals cluster depending on their sex or lifestyle. So the results, as shown here, show a strong correlation structure in the data. The various groups of males and females in different lifestyles tend to cluster together, indicating, again, a strong impact of both sex and lifestyle on the stress expression profiles of these individuals. And this is likely due to the shared environmental exposures. And of course, the sex effect is very, very clear in this data. So here, we did the same analysis, but focus only on males, labeled here as rural in blue, urban in red, and males who commute to the city in orange. So those are males who live in the village, but commute to the city for the day. And interestingly, the lifestyle transition pattern is readily visible in these profiles, with the commuting males being in a transition state. So our current work and future work will focus on other circulating biomarkers of stress. We are in the process of generating these biomarkers of stress in the serum of these individuals. We're also planning to do epigenetic profiling to test if the gene expression signatures that we see are epigenetically controlled. So as a summary, I hope I convinced you that lifestyles and environmental exposures have a very strong impact on gene expression profiles. I should mention or reiterate that we're talking about individuals who don't have any chronic diseases. Those are individuals who self-reported that they are in good health. We also show that the gene expression differences between lifestyles are likely impacting immune function and disease susceptibility, with urban individuals showing a higher level of biomarkers of disease. We also show that the urban lifestyle is associated with an increased burden of stress, both psychological and immune-related, as clearly shown in the gene expression profiles. We also demonstrated that genetic influences in our dataset are relatively minor, so differences between the two ethnicities, for example, within the same environment, are very, very minimal. Finally, I would like to thank our study participants and partners and collaborators in Morocco, and my team lab members who did the work, both in Morocco, as well as all of the downstream experiments in Abu Dhabi. Thank you.
Is it coming up? Oh, there we go. So I will be talking about agency and life satisfaction, framing local perception and tracing cause and effect through fuzzy cognitive mapping. Now, I'm speaking on behalf of my team research manager, Lina Qteshat, who wasn't able to attend. So I'm speaking on her behalf. She's the expert in this. And I'll start by giving an introduction. So I'm going to be talking about the intervention that I developed that I mentioned earlier, which is called We Love Reading. And its mission is changing mindsets through reading to nurture change makers. And if you want to know more about it, there's a book about it, which I'm also donating to the raffle for next year. What is it? What we do is we train women, men, and youth how to read aloud as an art and why reading for pleasure is important, and that they have to read in their native language in a public space in their local neighborhood. That's what it is. It's very, very simple. And the training is either in person or online. And we call them We Love Reading ambassadors. These are volunteers, ages 16 to 100. They go through stages of the training, from awareness to learning to fluency to mastery. And what we found is that the impact of these ambassadors reading aloud to the children was not only on the children, as the program, when it first was developed, improving their reading practices and attitudes, and also even reducing mental health burden and boosting resilience. But the surprising thing was the impact of the program on the adults, who were majority women. Helping them find their voices literally by reading and also figuratively becoming leaders in their community. And hence it became changing their mindset to a mindset of I can. And so this led us to, these are some statistics. The program now is in actually 69 countries around the world. We have more than 8,000 ambassadors all over the world, and you can see the rest of the numbers. The program has won multiple awards, and we have partnered with many international NGOs and local NGOs all over the world. Now, what we want to talk about in this talk is the impact of the program on the adults. And that was the surprising thing that wasn't planned for. But usually is the outcome when you do something organically and holistically, it's much better than when you design something on a table, especially if you're serving a community. So doing things from a local perspective is much more important. So we noticed in these adults who were reading aloud that from the anecdotal evidence that they were empowered and they were happier and they were doing better, and so on. So I was interested to measure that. Of course, it's not my field. So I invited scientists from different fields to do a randomized control trial on these women to measure. First was empowerment, as well as other things, but empowerment was my main goal. So in this study, we found life satisfaction was statistically significant, as you can see on the left side, but empowerment wasn't. And well-being was kind of on the threshold. I was very disappointed. And I thought, but wait a minute, I know these women are empowered, so what's wrong? And of course, my partner in this was Catherine Panterbrick, and she told me, Rana, you told me the surveys were fine, we translated them, they're culturally adapted. And I said, I don't know, Catherine, something's wrong. And so we went back and said, maybe those surveys that we were using that were trying to capture empowerment, although they were translated, culturally adapted, they were designed from a Western perspective. And we wanted to go back to the drawing board and ask these women, what does empowerment mean to them? Of course, that study, the randomized control trial, was published, but then we discovered a method called fuzzy cognitive mapping, which was suggested to us by Praveen Kumar from Boston College. And he said, Rana, this is the thing that you could use to capture what these women think about empowerment. So fuzzy cognitive mapping, from its name, it kind of captures what these women's impression, their mind maps of what empowerment meant to them. So we did it. It was fascinating. We published it as well. Now, what am I talking today is about, I love this tool. And so these women were telling me about how the program, We Love Reading, was improving their agency, their control over their lives, especially among the refugee population. Because when they're in like in a refugee camp, they're wandering aimlessly, they don't know what's going to happen, they're at the mercy of international NGOs or politics, and suddenly, when we introduced the We Love Reading program, it was like a secret sauce. Suddenly, all these women were fired up. They had a purpose. They were not just reading aloud to the kids. They discovered themselves. So they started creating their own programs, their own projects, finding a job, creating a business, embarking on a new endeavor. And to me, that's agency. That's that feeling of I can. I'm not a hopeless victim. So we said, let's use fuzzy cognitive mapping to study that. And that's what we did, and that's what this talk is about. So we wanted to focus on the local narratives to understand the complex concepts, which is the sense of agency and life satisfaction to determine the pathways of influence resulting from community-based interventions such as We Love Reading. So we asked these questions in color. What does a sense of agency mean to you? What does life satisfaction mean to you? What are the factors that impact these two constructs, and how those variables are interrelated in the system? And finally, how does We Love Reading as a program drive change, and to which aspects of the system? Now, the method that we use is fuzzy cognitive mapping, which is a type of causal system mapping used to generate mental models. It's a participatory approach. It represents and visualizes different stakeholders' knowledge, and it's a method to understand how and why complex systems change in relation to their own components. It's a semi-quantitative method and involves thematic analysis for stakeholders' discussion, and it also can be translated to numeric models of systems change. And you could actually run it and predict scenarios, as I will show you later. How it works, there's not much time to go through it, but the participants identify variables, and then they draw relationships between those variables, and then they specify whether the relationships are negative or positive, and then they assign relative weights to the causal relationships ranging from -1 to +1. And then we use the mental modeler online platform to actually feed in all this data from the session, and then come up with these really nice, cool maps. So we did this in Jordan. We did it in 4 groups: 2 Jordanian of this relatively same socioeconomic status, and 2 Syrian refugees. And these were conducted in December, from October to December of 2024. Now, each session was around 3 hours long. It was all in Arabic, of course. We recorded it all after getting permission and the IRB approval. And it took place in a community-based organization. And each session had roughly between 4 to 11 women. Now, the demographics of these participants, this is just a brief description, because this paper is not published yet. We're hopefully writing it up as we speak. So we had 31 female participants, half Jordanian, half Syrian. They were all We Love Reading ambassadors, meaning they took the training, they were reading aloud to the children. Their ages ranged from 16 to 60, again, typical of the program. And they came from diverse education backgrounds, from elementary school to PhD. And 36% had a bachelor's degree, 43 either completed secondary school or middle school. Only 1/3 of the participants had jobs, and 60% of them with full-time, of the 30 had full-time jobs, while the rest had part-time jobs. And 36.7% of the participants came from poor households earning less than 350 Jordan dinars, which is roughly equivalent to $500 per month. And 13% roughly of the participants earned more than 850 JDs. You can do the math what that means in US dollars. And this is just some pictures from the agenda and how it flowed. I'll share the results. So first of all, in the case of agency, the question of agency, the participants were able to identify total 12 meanings of agency, right? So, wow, compared to whatever agency metrics or surveys are being used today. So from their perspective, it was about freedom, responsibility, decision-making, control, independence, guardianship, leadership, self-confidence, privacy, giving, planning, awareness. And of course, this was, you draw it from all the quotes that these women were using. And in addition to the agency meanings, they identified multiple types of agency. So there was agency over people, over finances and resources, over actions, over body and wealth and health, agency over thoughts, over time, and over feelings. When we asked them about life satisfaction, it was about how they did the meanings of the concept were contentment, acceptance, belief in destiny and fate, peace of mind, gratitude, adaptation, realistic expectations, self-reconciliation, accomplishments, giving, compromise, and happiness. And then, and then they identified the different types of life satisfaction, from satisfaction with oneself, with health, with others, with trials and hardships, with sustenance, which is an Arabic word, arzaq, with life's reality and challenges. And when we put them together, we were able to see a core perceived meanings of both agency and life satisfaction together when we put the two constructs. And we showed that the notion of giving was kind of mentioned in both as a bridge between agency and life satisfaction, which is all interesting to delve deeper in, which we don't have time in this talk, but hopefully the paper will come out and you can look at it. Now, the participant women also suggested a sets of factors in these sessions, which they determined whether they were have positive or negative effects. And these were categorized either as personal assets, meaning the factors of money, education, and health, family and interpersonal, which included children, husband, relationships, family support, appreciation, approval of others, external interference, moral support, and then social and cultural category, which included environment, traditions and customs, structural and political, including rights, external circumstances, refugee status and security, and then spiritual, faith and belief and love. Now, we generated the maps. I'm showing you 2 of the 4 maps, and you can see how they cluster and how the variables are connected, and the thickness of the arrows and the direction of the arrows and the colors of the arrows indicate the different relationships, which I'm going to just briefly show. And in this way, you can look at the positive and the negative drivers and the system receivers. In this visual analysis, you can identify centrality of factors, centrality of agency, which we asked about, and the centrality of life satisfaction, and then look at what are those factors that were more influenced according to these concepts. So, for example, we found for the agency that the notions of responsibility, decision-making, control, privacy, and self-confidence were the most central dimensions of agency. While for life satisfaction, it was peace of mind, satisfaction with oneself, and the general life satisfaction were the central ones. Now, what we also did, again, we asked, well, how does We Love Reading impact the system? And so we found that We Love Reading was seen as a positive driver in the system, and that and as a central variable based on its perceived influence by the participants, starting from an interpersonal level. So it was the engagement of the ambassadors in reading aloud with the children, with the families of the children, and the wider community. And so the respondents linked the We Love Reading program in this way to the notion of giving. We also, at the personal level versus the interpersonal earlier, that it was this being in charge of decisions related to conducting the reading aloud sessions, like when they were, the timing, the frequency, the location, the storybook choice, and recognizing their positive influence. It enhanced the We Love Reading's ambassadors' satisfaction with themselves and increased their self-confidence. And as you can see that in these maps. Then we did 2 scenarios of change. So what that is that you say, what if? So you run the prediction. And what we did is the aim of this step was to trace the cascading effect of the program once activated to its maximum value. So in these scenarios below, among the dimensions of agency and life satisfaction, the level of responsibility and satisfaction with oneself are predicted to receive the strongest positive influence. And interestingly, the cascading effect of We Love Reading on agency indicates a positive change in notions of control across all 4 maps. And so we compared these results with an earlier study we had done on Emirati women from the UAE. And the difference between the Emirati women, they both took the program compared to the Syrian and Jordanian, but the Emirati women come from a very high socioeconomic status. Very, very high. And so we found that we did the same agency mapping, and it was similar across all these Arab women, regardless of your socioeconomic status. And this was published recently, and this was work done with Catherine Panterbrick and Dina Tbeshat, as well as Lena and Yannick Egerman. And so this led us to kind of We Love Reading's theory of change, that it's about reading aloud. When the women are reading aloud to the children, the child, and that's a whole different set of research, is changed, but the woman, the adult, also gains her self-confidence, her sense of ownership and agency, and her well-being that results in her becoming a change maker. And of course, I have to tie it to my biology as an epigenetics person. So actually, we finished collecting all the data from a randomized control trial on the women who read aloud and the children between Syrian refugees and Jordanians. And we are going to look at the pediatric epigenetic clock with Michael Kober to see if we can find an epigenetic signature shift by the children who are exposed to trauma, the Syrians compared to the Jordanians, and then seeing after reading aloud for 3 months, do we see a shift? We already know that there's a mental health shift, but we want to see if there's a shift in the children and in the woman reading aloud, and if there's a mirroring. So stay tuned for that in the next few years. So my recommendations, all of us together from this research, is that we need to evaluate the perceived benefits of community-based programs, such as We Love Reading, through the eyes of the volunteers. because it's crucial for testing assumptions about local knowledge and the transformative power of these interventions. Second, this research demonstrates participatory approaches like fuzzy cognitive mapping is important to unpack the local knowledge and systematically explore causal reasoning and systems change, and actually to draw new surveys from these fuzzy cognitive mapping. Other programs aspire for a long-term sustainability, can benefit greatly from understanding what motivates their volunteers. Investing in strategies that balance volunteers' motivation and commitment can help ensure overall success. And successfully implementing interventions in low and middle income countries requires tailoring programs to the local cultural context rather than simply translating existing models. And lastly, adopting community-based programs like We Love Reading by the family, community, and at a national level where reading for self and reading aloud for children becomes a strategy and a cultural habit is essential for human flourishing and well-being. We've created a campaign to create a 1 million We Love Reading ambassadors, and our star is Esma, and there's a whole documentary done about her, which is free, so you can scan the QR code and join the campaign. And finally, I want to acknowledge all the We Love Reading ambassadors who have participated in this study all over the world, and the funder, which is the UNESCO office from the government of Korea, South Korea, because we won the International UNESCO Prize. And you can know more about We Love Reading through all our social media and website. And we have some children's books here for take, which I'll put over there, that we produce to enrich the literature that is being used for the children. And with that, thank you very much for listening.
Good morning, everyone. My name is Sarah Merrill, and I am an assistant professor at the University of Massachusetts Lowell studying social epigenetics. My colleagues and I are excited to share with you our work on the effect of a resilience-building intervention on epigenetic age acceleration of Syrian and Jordanian adolescents living in Jordan. Early life adversity is a global health crisis, predisposing those who experience it, and especially those who experience it at higher rates, to a litany of potential health risks in the future. One potential mechanism by which this may be occurring is biological embedding, or the process by which experiences can get under the skin to alter biological processes. For example, adverse experiences may lead to adaptive biological changes, which in turn may lead to different developmental states. This is one way in which early life adverse experiences may be affecting later life development and health outcomes as mediated by these dysregulated biological responses. One potential biological mechanism by which this could be occurring at the intersection of genes and environment is the process of epigenetics. Epigenetics are the persistent, mitotically heritable alterations that do not change the underlying DNA sequence. Social epigenetics is the process by which life experiences can affect biological pathways, which can in turn influence how our genes are expressed. This can result in different phenotypes, such as differences in health, and subsequently may influence various biological pathways in response. While there are multiple epigenetic modifications, today we will be discussing DNA methylation, which is the deposition of methyl groups onto the genome at specific sites. One way to think of this is to consider the genotype you are born with as a window of potential expression. DNA methylation can then potentially act as a dimmer switch, adapting expression of a gene, given the limitations of your genotype, and as influenced by your environment, such as the experiences of trauma or early life adversity. A key feature of DNA methylation is that DNA methylation at specific sites in the genome change reliably with age across individuals. Therefore, by measuring DNA methylation at these particular sites, we can estimate the approximate age of someone based on their epigenome. This is called biological age, or the prediction of an individual's chronological age representing their functioning, growth, and health. Epigenetic age is a type of biological age, specifically using DNA methylation markers as predictors. Epigenetic age acceleration is a measure of how fast an individual is aging relative to their chronological age. Therefore, someone who is estimated to be epigenetically older based on their DNA methylation than their chronological age based on their date of birth would be experiencing increased epigenetic age acceleration. In adults, a well-replicated and validated finding is that increased epigenetic age acceleration, again aging faster epigenetically than chronologically, as illustrated by the individual in red here, is associated with poor health outcomes, including increased risk for disease, frailty, and even mortality. However, it is less clear what epigenetic age acceleration means in children and adolescents, though work from the past decade has repeatedly found associations of early life adversity such as trauma, abuse, deprivation, and PTSD with increased epigenetic age acceleration in children and adolescents, and even retrospectively in adulthood and older adulthood. As such, it is critical to understand how intervention, especially accessible, evidence-based psychosocial interventions, could act to buffer or even prevent epigenetic changes to adversity, such as epigenetic age acceleration, to prevent future health and development differences. One such evidence-based and resilience-building intervention is advancing adolescence. Advancing Adolescence is part of the No Lost Generation program implemented with war-affected youth, specifically in our study, Syrian urban refugees and Jordanian hosts. The focus of Advancing Adolescence is to alleviate toxic stress, build resilience, and create support. The program aims to do this in three main ways: the profound stress and attunement framework for addressing feelings of depression, anxiety, and vulnerability, and processing experiences of violence, discrimination, isolation, and uncertainty. The community-based protection network for helping to build social capital, creating social cohesion, and building systems for community-based organization. And third, the education and life skills building, connecting adolescents to coaches and mentors, organizing activities, and building vocational skills and opportunities. This program has already been found to improve social cohesion and stability, feelings of safety, and confidence in the future among adolescents. Additionally, Drs. Panter-Brick and Dejani have published on the benefits to mental health and biology found in response to intervention within our cohort. Specifically, they found participating in the Advancing Adolescence intervention reduced feelings of distress, insecurity, stress, and improved mental health. They also found a reduction in hair cortisol concentrations, a stress biomarker, after intervention. Given these psychosocial and biological effects the intervention previously discovered, we hypothesized that the effect of advancing adolescence may be observed in epigenetic age acceleration changes. The main goal of our analyses were to examine whether advancing adolescence reduces epigenetic age acceleration, determine whether displacement and trauma exposure levels affect intervention effectiveness, and assess whether intervention effects persist long-term. Dr. Rana Dejani and Catherine Panter-Brick allowed for us to characterize epigenetic age at 3 time points using waitlist randomized control design. Not all participants who received the intervention had DNA methylation, thus leading this to be a quasi-experimental design but based on an RCT foundation. In total, 175 Jordanian and Syrian adolescents from 11 to 17 years of age gave cheek swab samples and questionnaires at 3 time points: before intervention, 10 weeks later, immediately post the 8-week Advancing Adolescence intervention, and almost a year later. There were equal numbers of boys and girls, and all adolescents had some level of identified mental health and/or behavioral risk, with the Syrian displaced adolescents reporting 1 or more traumatic experiences at baseline. Within the samples, we examined 2 appropriate epigenetic clocks: the Horvath pan-tissue PC clock, for which predicted ages were correlated 0.56, which is similar to other reported cohorts, and the PED-BE, or pediatric buccal epithelial clock, which is also an appropriate tool. This clock had similar but slightly higher correlation of 0.67 between the chronological age and predicted epigenetic age of the children. Epigenetic age acceleration was calculated as the residual to the line of this regression, correcting for estimated buccal epithelial proportion. Cell type, sex, and age at sample collection were accounted for in all models. First, we began with examining the effect of intervention for the entire group with the PED-BE clock. We observed an increase in epigenetic age acceleration in the control group at T2, our 10-week post-intervention time point, and a slight decrease in our intervention group. This was a statistically significant difference at T2, meaning the intervention group had lower epigenetic age acceleration than the control group. This suggests that the intervention may have had an initial protective effect, preventing the increase in epigenetic age acceleration seen in the control group. We see a similar pattern using our other clock, the Horvath pan-tissue PC clock. Here we see the same effect, but stronger. While the initial intervention effects from T1 to T2 seem protective, by T3, our 11 months later time point, epigenetic age acceleration increases to match the control group. This is potentially due to a time-limited effect where intervention only temporarily buffered biological aging, but did not provide lasting protection. There are a few explanations for why this outcome may be. One is where the intervention may have delayed the effects of risk and adversity, but not eliminated them. There could be other external factors or ongoing stressors that may have driven epigenetic age acceleration increases in the long term, potentially because of being without the continued intervention support. Or this could also be evidence of a stress recovery mechanism, specifically a rebound effect in biological aging seen in the long term. We examined this further with specific subgroups. When examining if intervention effects were similar for those reporting high, low, or no trauma at baseline, we observed differing patterns. However, with the PED-BE clock, while there was an increase in epigenetic age acceleration over time, especially for those reporting high trauma, regardless of intervention randomization, no other differences were statistically significant. However, the same pattern was observed in the Horvath pan tissue clock. In this clock, we observed, similar to the group level comparisons, the control group has significantly increased epigenetic age acceleration compared to the intervention group at T2. This was a slight difference in the high trauma group. There was no difference in the low trauma group, and a large significant difference in the no trauma group. Particularly in the no trauma comparison, the control group's large epigenetic age acceleration increased at T2 suggests external factors that the intervention may have buffered against. This suggests that the intervention was effective at T2 in the short term in slowing the acceleration of biological aging, but again by T3 the protective effect seems to have worn off as the intervention group catches up. However, it could be that those reporting traumatic events, both the number and the scope, are quite different. Specifically, we looked at the number of traumatic experiences endorsed at baseline for the displaced Syrian adolescents and the host Jordanian adolescents in this violin plot. Syrian participants reported significantly higher trauma exposure than Jordanian participants. The Jordanian adolescents' trauma scores were mostly concentrated below 5, with very few higher values. These findings align with the expected patterns, as Syrian participants had more conflict-related trauma exposure, whereas Jordanians were in a relatively more stable setting, reporting lower overall trauma levels. This elucidates our results when examining our data by displacement, with Jordanian adolescents again showing that the intervention initially significantly reduced epigenetic age acceleration at T2 and lost effectiveness by T3. However, for the Syrian adolescents, both the control and intervention groups showed similar trajectories of increased epigenetic age acceleration over time, potentially due to the more extreme conflict-related trauma the Syrian adolescents had already experienced. These mirror the results from the no trauma and high trauma groups, respectively, we observed earlier. The same pattern was observed with the Horvath pan tissue clock, though again with a stronger effective intervention from T1 to T2. The observed differences may be due to Syrian adolescents experiencing significantly higher trauma, which may make epigenetic age acceleration trajectories less influenceable. There may be other contextual factors as well, such as resettlement, social environment, economic security, and healthcare access, that act as ongoing stressors that counteract the post-intervention effects seen at T2 for the no trauma and Jordanian adolescent groups. It is also possible that there are simply heterogeneous responses to intervention, and some individuals may be biologically responding better to the intervention than others. Future research will break down the facets of intervention effectiveness, rather than randomization, that might be most strongly influencing epigenetic age acceleration, such as reductions in inflammation, stress biomarkers, and mental health scores in these adolescents. Some key takeaways from this study are that high trauma exposure is linked to increased epigenetic age acceleration, which is similar to what has been seen in previous studies, and that this is most evident over time. Syrian displaced adolescents reported significantly higher trauma exposure than the Jordanian adolescents, and that higher trauma exposure may have affected intervention efficacy, at least in terms of biological aging. The intervention significantly reduced adolescent epigenetic age acceleration at that T2 post-intervention time point, and this was primarily driven by the Jordanian adolescent no trauma group. A year later, intervention and control groups converged. We did not observe any lasting protection for biological aging once the intervention support ends in any group, and trauma level and displacement influenced the intervention effectiveness in terms of buffering biological aging. Specifically, Jordanians and those with low trauma appeared to benefit more in the short term, and contextual and environmental factors such as housing or socioeconomic instability may buffer intervention effects. Thank you so much to everyone who made this work possible. my colleagues, Dr. Rana Dajani and Catherine Panter-Brick, as well as Dr. Michael Kobor, and all of the families and adolescents that made this work possible.
But we will go now to our last speaker, who will join us from Palestine. She'll be it's not recorded. She'll be presenting live via Zoom. Her name is Haneen Dweib. She's an assistant professor at Birzeit University, and she'll be talking about sexual dimorphism of the fasting adipose, mitigation of metabolic and adipose tissue, mitigation of metabolic and cardiovascular dysfunction in a non-obese pre-diabetic rat model. It's not a human model, but the person, somebody was going to present, but they weren't able to present. So Haneen stepped in to connect the rat model with the fasting in humans and Palestine. So Haneen, the floor is yours. Please go ahead.
Hi, thank you, Ren, for the introduction. Apologies in advance for my voice, but I'm really sick, so I hope that you can hear me well. So my name is Haneen. Today I'm going to present about the sexual dimorphism of the fasting adipose. But before we start, I'd like to give you a disclaimer about my presentation. So this is not a political talk. I'm not a politician. Neither am I affiliated with any political party or ideology. I'm just a human, a scientist, and a proud Palestinian woman. Saying that, I can move to give you a brief background of how science and life behind the wall that separates West Bank and Gaza from the rest of Palestine is about, because it's important to understand the context of how we're doing all of it. Palestine now is separated into two main parts, Gaza Strip and the West Bank. And you may think that this wall is like really old, but this is a product of the millennium. It's not like the Berlin Wall. I don't know why my laptop is very excited to move on to slides. So to give you a bit of context of how things work and how Palestine is segregated and confined in these two small areas. We have Gaza Strip, which is here, and we have the West Bank. The West Bank is divided into three main areas: Area A, B, and C, and the fourth area is H2, which is specifically in the old city of Hebron. So not only the locationally or geographically Palestinians are divided, but also in terms of IDs. So we have two main IDs. We have the green ID and the blue ID. So what does that mean, and how does it reflect on the daily lives of Palestinians? So for West Bankers, the green ID only gives them access to West Bank areas. As for Gaza and Jerusalem, they need special permits. So for Gazans who have the same ID, which is the Palestinian ID or the green ID, they only have access to Gaza, and they had to get special permit to go to West Bank and special permit to go to Gaza. The blue ID on the other side, I don't know what's happening. The blue ID on the other side, it's for Palestinians residing in Jerusalem, and they have freedom of they can move freely between West Bank, Israel, and they should have special permit to Gaza. So just I have to point out that Palestinians on both sides of the wall are 100% subjected to limitation and restrictions of movement. So not only the IDs, but also the borders. So we have Gaza, they have access point to elsewhere in the universe through Egypt and Rafah. I'm sure that you've heard about it in the last year. For West Bank and Jerusalem, we access Jordan through Jericho. So the thing is that we don't have an airport. So we have to travel twice. We have to travel to these countries. Like if you are from Gaza, you have to travel to Egypt to access the airport. If you are from West Bank and Jerusalem, you have to travel to Jordan to access the airport. And these are not like the most humane circumstances. Like these land borders are not the best to travel. especially for Palestinians, not for foreigners. So now I'll take you with me to a regular trip that I used to make like couple of times a year from Palestine, Bethlehem to Amman, because Bethlehem is my city. And I travel, like if I want to go to the States, I travel to Amman, and then from there I'll take like the flight. So from Bethlehem, I ride on a bus that will take me to the Israeli borders. And then the Israeli borders actually is the bottleneck of the whole process. And then you get multiple security check, baggage check, and stuff. And then you change the bus to go to the Jordanian border. And then you have another security check, baggage check, and stuff. And then this process, like sometimes it take literally like 45 minutes. Sometimes it takes 6-7 hours. Sometimes it takes days. Like the trip I did to Muscat like last month, it took us 2 days to get to Muscat because like we had to travel in advance, like 1 or 2 days in advance. So what about life in West Bank? You might wonder how life is in that area. And I'm going to tell you it's not that exciting. We don't have much of freedom, as you can imagine. We don't have freedom of speech, neither movement, transportation, traveling. We have work limitation, study limitation. We have horrible internet connection. I'm so happy you can hear me now. And we pay taxes like 3 times on goods, fuel, water, electricity, on everything. And we have apartheid and war and constant occupation. So what about research? Because like you can imagine, like in war zones, we don't have the priority to have like biomedical research or clinical research or like more advanced research. It's like it's very limited, funds are limited, research isn't a priority, and the infrastructure is very poor and primal. Like we don't have an advanced infrastructure. And I did like the first clinical study at my university, which is Ahliya, Palestine Ahliya University. And that research was mainly funded by me and the university. And I borrowed a bench at a medical lab, which is a friend of mine has this lab. So you can imagine how things are done. Plus, we don't have movement restrictions. So if I'm collaborating with Birzeit University, which is like should be like half an hour drive, it might take like 3 days to get there. It's very expensive to us, and it's a constant war zone. We don't have anything stable. One day we are like living normally, the next day like everything is shut down. So before I move on to the science part of this presentation, I'd like to share some of the experiences of my students during the Gaza war, because I had the honor to teach some of these students on a special program that universities in West Bank, they accommodated thousands of students from Gaza, but unfortunately, we don't know anything about them now. So the first one is Diana. Diana, she once wrote to me that, and these are screenshots approved by these students to be shared in here. These are screenshots of the conversations from last year. So she was telling, like, I'm writing to inform you that due to the current circumstances and the impeding occupation attack on Rafah, we were forced to evacuate and return to Khan Yunis. Unfortunately, the Wi-Fi over there is unavailable at the moment. I might not be able to attend this class. So she was explaining why she's not attending, although they were like at a constant bombarding and carpet bombing. And she was she was also explaining that she was always the top student, but this security situation halted all of our progress and dreams of education, which is really sad. The other student is Amna. Amna was contacting me after the midterm exams because she didn't do very well, and she explained her story. So I asked Amna to tell you her story with a video. But before that, she said, like, when I initially opened the camera to record, these are the exact words of Amna. When I initially opened the camera to record my experience and suffering during the war, I realized that it was the first time I was truly facing my feelings. The emotions and memories overwhelmed me, leading me to record multiple videos, but ultimately deleting them. It took me some time to gather the courage to confront my suffer and capture it on a video. Now I leave you. We will wrap up this part with the words of Amna, and I guess they are, I believe that they are really worth to be heard.
In the wake of war's devastation, I found myself shattered, losing not just my husband but also beloved family and friends. My world crumbled, passion extinguished by grief, drowning in the torment of loss and the ever-present specter of fear. But amidst the darkness, a flicker of hope emerged. I sought refuge in education, a beacon guiding me through the abyss of despair. Dr. Hanin's compassionate concern fueled my determination, igniting a newfound purpose. With each step forward in my educational journey, I defy the shadows of suffering, reclaiming my strength and resilience. This is my story of turning tragedy into triumph, of finding light in the darkest of times.
So Amna lost 23 family members, among them her husband, and she was forced to leave to Egypt and to work full-time and to study full-time either as well. Unfortunately, I don't know anything about Amna or Diana or the other students because like we lost contact with them. So with that part, I start the other section of my presentation, which is about the sexual dimorphism of the fasting adipose. So according to CDC, 1 in 3 Americans have prediabetes, and 80% they do not know that they have it. So why we are determined to study the sex differences in metabolic and cardiovascular dysfunction? Because women are more insulin sensitive, they have less adipose inflammation, and they have more resilient and adaptive mitochondria. And these, the advantage, the metabolic advantage of women is usually based on the anatomical differences by sex hormone and adiposity patterns, as you can see. But how about the perivascular adipose tissue, which is a thin layer of adipose tissue surrounding the vascular beds, and which is the tissue of interest? We haven't found so many about sex differences and PVAD in response to a high-fat diet or hormonal therapy or whatsoever. So on the animal side, we found that animal studies diet-induced PFAD dysfunction was more prominent in male, and ovarectomy-induced PFAD dysfunction as well. In human studies, we found very scarce studies, but one of them stated that postmenopausal women had a higher mass of PFAT inflammatory and inflammatory marker compared to premenopausal women. So in this graphical, in this figure, you can see that how from our work that positive energy intake will lead to metabolic dysfunction and insulin resistance through PFAT inflammation or dysfunction, while presented by hypertrophied adipocytes and hypoxia, increase in hypoxia and subsequent inflammation. So from our previous findings, hyperinsulinemia led to PVAT inflammation and early cardiovascular dysfunction in this pre-diabetic animal model. This PVAT inflammation was fueled by increased or hypertrophied adipocytes and upregulation of uncoupling protein 1, which will lead to a hypoxia that eventually will fuel inflammation and PVAT. The pre-diabetic rat model is based on Sprague-Dowley rats. Initially, we started with male rats that are fed for 12 hours. And eventually, we will have a non-obese pre-diabetic model with phenotype of apologies. Normal weight, that they have normal weight, but they have increased adiposity, hyperinsulinemia, normoglycemia, and insulin resistance compared to the control rats. So we hypothesized that isocaloric therapeutic fasting, daily isocaloric therapeutic fasting, will improve insulin levels and sensitivity, will lead to positive remodeling of PVAD and improve cardiovascular function. For that purpose, we randomly separated the rats into, we started with male rats for 3 main groups. The first one, they will eat normal chow for 24 weeks. The second group will eat hypercaloric diet for another 24 weeks. And the third group where we established the insult by 12 weeks of hypercaloric diet. And then we started a daily fasting for 12 hours. This fasting regime is during the dark period where they are not 12 hours daily fasting. They are not allowed to consume any food, but they have free access to water. During the light period, they will have freely access to the same diet. So interestingly, therapeutic fasting corrected pre-diabetic metabolic phenotype in these rats. As you can see here, that they are not obese, but they have increased adiposity. They have normoglycemia, but they have increased insulin level or hyperinsulinemia with an increase in insulin resistance that all were corrected with therapeutic fasting, isocaloric therapeutic fasting. So moving to our tissue of interest, which is the perivascular adipose tissue, as you can see here, the adipocyte size was, the hypertrophy was rescued by intermittent fasting or therapeutic fasting. Also, the hallmarks of PVAD dysfunction, they were all corrected, which are the UCP1, which is the uncoupling protein 1, also GRP1, which is a biomarker of mitochondrial fission, and also it corrected hypoxia measured by hypoxia-inducible factor 1 alpha, and eventually the inflammation. So all hallmarks of PVAD dysfunction were alleviated by therapeutic fasting. It also improved the baroreceptor sensitivity and improved by the fasting regime, as you can see here. So to move on to the sex differences part, we had intact females, Bragdowli females, with the same exact groups as the males. So as expected, the metabolic phenotype of intact females did not give us any type of prediabetes. Although they presented hypertrophy in PVAD adipocytes, it was corrected with fasting. There was an upregulation of uncoupling protein 1, but it wasn't enough to induce or elicit any of the dysfunction markers, which suggest the role of estrogen in preventing the protective role of estrogen in preventing early cardiometabolic disorders and prediabetes. And for this purpose, to study the role of estrogen, we added another batch with the exact diet groups, but these rats were ovariectomized. Okay. So we had these female rats ovariectomized at week 12. And then moving on, we either continued on the same diet or introduced the therapeutic arm. So to our surprise, ovariectomy induced obese prediabetic phenotype, which is worse than the male phenotype, prediabetic phenotype. So they were obese, they had prediabetes, and which was all corrected by fasting alone without any estrogen therapy. So as for the PVAD dysfunction hallmarks, the adipocyte size were getting smaller with the fasting regime, and the markers of PVAD dysfunction, UCP1, HEF1 alpha, and IL-1 beta, they were all reduced by the therapeutic fasting per se. It also improved the baroreceptor sensitivity. So taken all together, isocaloric fasting improved hyperinsulinemia, and hyperinsulinemia reduced the hypertrophy of PVAD of adipocytes and PVAD, which improved the oxygen supply. And on the other hand, hyperinsulinemia improved or reduced mitochondrial fission and reduced free fatty acids. And then both reduce the activity and expression of uncoupling protein 1, leading to lowering further the oxygen consumption, both lowering the oxygen consumption and the increase of oxygen supply, improve the oxygenation state of this tissue, which led to a state of normoxia and PVAD. Normoxia is a state of anti-inflammation that eventually improved cardiovascular function. So our take-home messages is that PVAD plays a role in modulating cardio and metabolic dysfunction in early prediabetes in a sex-dependent manner, despite being a very minute adipose pool. Isocaloric fasting risked from metabolic and cardiovascular impairment in pre-diabetic males and ovarectomized female rats. And this fasting regime can be adopted easily to correct prediabetes and its complications in men and postmenopausal women. So with this slide, I'd like to thank you for listening and for your time. I'd like to thank all the people in this slide, especially my husband and my daughter Sama, for their continuous support and love, and all the collaborators on ISCA for Women in Science and the O's for their generous support. Thank you so much for listening, and please do not hesitate to contact me if you have any ideas to collaborations or if you have any questions. Thank you so much.
Thank you, Haneen, and I hope you feel better. So with that, we concluded our 6 sessions and talks, and I just want to highlight how all these projects, some of them are related, the importance of establishing relationships, international and local, and how good collaborations and good relationships matched with good questions. How do you ask the question? That's the most important thing, because when you put those two together, you do better science, and science that keeps forthcoming, because you ask one question, you get 10 more, and you keep on going and going and going, such as the research that Catherine and I have been working on for 10 years, and it's still producing results and new papers. So thank you for all the speakers, and thank you for everyone for being here and staying for the last day and the last session of the conference. Now we know we're here till 11. There's supposed to be coffee outside. And I have a box of sweets from Jordan, because I came from Jordan. So these are Arabic baklava, one of the best. So please have it with your coffee. But until we break for coffee, we're going to open it for more questions. So if anybody has a question, all our speakers should be on Zoom and should be able to listen, I'm assuming. And Fatin and I are here in person. So any questions, please go ahead. Oh. And it's okay if you don't have questions, because you can always email us later. It's a lot of information in a very short time. And we appreciate you listening and enjoying, hopefully, the talks.
So, well, I'd like to say I really appreciate the combination of talking about the context of science as well as the actual science being done. And I always come home from India or from Mexico or wherever I'm working. I always come home so inspired by people who are doing so much with so little, in terms of the money that is available and the resources and the internet and the things that we take for granted. And that right now in the political situation here, I think that we're going to get a dose of harsh, I don't know what, I don't want to say reality, but it's going to be a harsh surprise of how to do science whenever we don't have easy access, not that it's that easy, to money and resources. And I just think that what you are presenting to us is so important for us to take away, because it really is the science, keeping the science front and center, even if the resources aren't there. And I just really appreciated that combination. Thank you.
Thank you. Thank you. I couldn't agree more. There's so much we can do together by sharing and learning. Any other questions? So maybe I'll just throw out, like, there are challenges, as you said, there are the typical challenges that were mentioned, but there are other challenges that were not mentioned, like language. Like the Arab world, we all speak Arabic, that's the native language, and science is spoken and written in English, and that provides a barrier. And therefore, this translation was very important. And hopefully these talks will be online. Are they going to be shared? I don't know. That would be great for our students to use them in our teaching. And so language is important. And maybe I'm going to throw out, maybe at some point in the future of the HBA, you'll have a regional conference in the Middle East. And then we could have a lot more going on and a lot more learning as well. So we have to dream.
So regarding the language issue, is AI helping with that? Are you using it for publishing? Are you using it more for live translations, things like that?
Yeah, so I think it's more for live translation, especially when it's live or when you share a presentation, so that the students can really comprehend what the speaker is saying. So they read English and the science, but when a speaker is speaking fast, they're not going to absorb as much as when it's translated into their native language. And then if it's recorded, they can go back to it later at their leisure and look at it. So AI is being used for that. I think the translation a lot. There's been an argument within the Arab world because I think around 400 million people speak Arabic across the Arabic world. Is there a way, because in China, for example, everything is translated into Chinese. Not every Chinese reads all the English literature. There's a governmental effort to translate. I think similarly in Japan. And so the question, which actually we're debating in the Arab world, should we do that? But then that would require a very huge collective effort across all the Arab region to put in money to translate. And that way you raise the level and the bar of scientific knowledge and awareness when they're reading it in their own language. And I think AI can step in here and help in that. Translating textbooks, for example, that's one thing that can be done easily. Scientific journals, that's a little bit more difficult. But again, AI can help in doing it quickly. So there are a lot of advantages to AI. It's just how you use them. Yes.
I mean, following on that, I think our work as society has to convince journals that it's worthwhile accepting translations. I had an experience of a paper we published in our journal, American Journal of Human Biology, just after the pandemic. Well, in collaboration with other co-authors, of course. And I translated the paper into Spanish, and originally they allowed us to put it as supplemental material, and it went all well. And then several months later, they took it down.
Oh.
So, I mean, this is costing nothing to the publisher because the translation was our responsibility. We double-checked that it was coherent with the science. You know, I'm a native speaker.
Yeah.
And nevertheless, they took it down. So I think that's an opportunity for us to push towards it. We're not asking the publisher to take the cost or time or effort, but to allow us to have a space within the framework of online publishing for authors who want to expand their reach to take care of the publication in their own languages. And that's a start, but we really need to push towards that.
Oh, that I couldn't agree more. I echo you because we can translate and people are ready to volunteer to translate if somebody's willing to publish. And it will not just enrich the speakers of Arabic, it will enrich the world because it goes both ways. A lot of publications in the Arab world are published in Arabic, especially in the social sciences. They're all in Arabic. And that is inaccessible to the whole world. And so and that limits the knowledge and the diversity of knowledge that it's important for us to keep on pushing science forward. So it works both ways. If we can translate the Arabic into English and the English into Arabic, then we're enriching the world. So I support that. And if we can help the HBA or whoever, that would be great. All right, we don't want to stand between you and the coffee and the sweets and the rest of the day. It's been a pleasure organizing it, working with the HBA team and all the speakers. Thank you, and all the listeners, whoever's online, and all of you here. Thank you very, very much. And if you ever come to Jordan, you know someone now, and you know a lot of people through the speakers. Thank you very much.